The overall goal of this experiment is to characterize the evolutionary pathway of antibiotic resistance using a morbidostat that continuously monitors bacterial growth and dynamically adjusts the antibiotic concentration as bacteria acquire drug resistance.
The main advantage of this technique is that the setup is made of the low-cost, easily available electronic components and can be easily reconfigured for specific needs. Demonstrating the procedures will be Po-Chun Liu and Yi-Tai Li. Both are grad students from my laboratory.
To assemble the morbidostat, begin by using an 18-gauge needle to punch 3 holes into the cap of the culture vial. Cut 3 pieces of polyethylene tubing approximately 7 centimeters in length and insert them into the holes in the cap. Use tape to wrap the edge of the cap to serve as the cast for the polydimethylsiloxane, or PDMS, mixture. Then add 5 grams of component A and 0.5 grams of component B of the PDMS in a 150 milliliter plastic container and use a toothpick to manually stir it. Load the mixture into a 10 milliliter syringe. Then, with the syringe, inject the PDMS mixture onto the cap. Bake the entire cap at 70 degrees Celsius for 8 hours to cure the PDMS. Next, using a soldering iron, solder the LED anode with the 24-gauge wire and the LED cathode with the carbon resistor. Then, solder a 680 ohm